I changed my title a bit, but I mean, first of all, maybe I should apologize to people who heard me talk, I think, across the road from where you are today in December, whoops, or November. The beginning will be slightly similar, but then will quickly diverge. So I'll taste of sort of quantitative work we've been doing in immune repertoires with my long-term collaborator Thierry Modar here in Paris, and a lot of collaborators both locally and experimental and theoretical people from all over the world. So I'll thank them to begin with. All right, with that, I'll start with a whodunit. So instead of being sort of in the center of universe as you are right now, imagine you're in a remote location, everybody's excited, there's an amazing after party, but the next day Joe is found dead. And next to Joe's body, there are a few drops of blood that don't belong to them. So this being scientists, they cry genetic tests for everyone, and traditionally used tests in DNA tests in forensics reveal that the murderer is one of two twins, either Alex or Terry. And so traditionally used tests in forensics are not able to distinguish between identical twins. Of course, you being where you are, you probably know that there are signatures in the DNA that are able to distinguish, but that's not what's being used. But here we're going to call in Detective Asia Bear, who is going to use another technique based on immune repertoires. So immune repertoires, basically B-cell and T-cell repertoires, are what we call a diverse ensemble of set of cells whose role is to protect you against foreign pathogens. And so what this set of cells is able to do, and what each one of these cells is in principle able to do, is to bind proteins, different proteins that are presented to it, that it encounters, and perform a discrimination task. So basically figure out whether this cell is natural to your body, your own self, what we'll call a self protein, or if it's foreign, in which case you should get rid of it. And so if it recognizes a protein as foreign, then it'll proliferate. It'll make many copies of itself that will all have the same receptor as the original cell. If it doesn't recognize anything, after a while, it'll die. And the thing to realize here is that while each cell has many, many receptors, they are all of the same kind. They're all identical receptors. So if you want to protect you, if you need to protect yourself against many different pathogens, you need a diverse set of different cells, and that's what we call a repertoire. And that's also why when they proliferate, they keep the same receptor. So this is the story for T cells. I'll be jumping a little bit. I'll mostly be talking about T cells, but I'll be jumping a little bit between B cells and T cells. I'll tell you when things are a little bit different and when that's important. But in both cases, we can now go in, take blood from people, sequence it, pull out the T cells or B cells, sequence them. And since these are proteins, what we're going to get is we're going to get reads that actually encode for this protein sequence. So in principle, what we have here is we have access to your own personalized medical record, because because of this proliferation process, the immune system keeps track of every infection you had. So if we knew how to read what we get out of these sequences in a functional sense, if we knew how to relate this to antigens or diseases, we would know in principle every disease, or at least signatures for the diseases or conditions that you carry still in your blood. But unfortunately, we don't currently know how to read it completely. But basically, this is what I'm interested in doing. I'm interested in figuring out how to read this information that we carry around in ourselves. So how will this help us find the murderer? Well, so if we compare the sample of blood found by Joe's body to samples from our two suspects, we can simply count the number of shared sequences between the two samples. And based on this, we can figure out whether the two samples come from the same person or whether they come from two different people. this is what the data looks like. So if the samples come from the same person, they are going to share many more sequences, an order of magnitude more sequences than samples that come from two different people. Even if the two different people are identical twins, what we see here in pink. So basically, you share more T cell receptors with yourself than with anybody else in the world, even if you have an identical twin. And so thanks to that, we'll be able to identify that the murderer is Alex, because who else could it be in this story? And additionally, if we found this drop of blood later in time, or we did the analysis later in time, we could still find the murderer. The signal is stable here. We show it based on data of days post vaccination. We're able to, the people look different on the order of days, but if we use a model, this can extend up to years. And Andrea will show you how we find these models that are able to extend things over the years. But at this point, you may be asking, but how does this work in detail? So it works. So I'll explain how we can figure it out. But first, I need to sort of tell you a little bit more about how a T cell receptor is made. So this is the classical textbook picture. A T cell receptor has two arms, what are called an alpha and a beta arm. Let's just concentrate on the beta. They're made out of 3 segments, which are called V, D, and J. And in your genome, you have a certain number of templates for V, D, and J. Okay, so maybe the first thing to realize is, before we go into these details, is that you have of the order of 10 to the 9th unique or different T cell receptors, at least. Now, you have of the order of 20,000 genes. So since these are proteins, if they were encoded directly in your genome, simply the DNA wouldn't fit into your nucleus. So we need a different procedure, and this procedure combines combinatorics and randomness to generate this diversity. So in each of your cells, you have a certain number of templates, but then the DNA in your cells gets edited. So you pick one V, one D, and one J in each cell, which gets you to the order of 1,000 different combinations, but we need to get to 10 to the 9th. So to do that, there's enzymes that come in and randomly insert and delete nucleotides at the junctions between the V and the D and the D and the J. And with these insertions and deletions, we're going to get to this 10 to the 9th, or otherwise said, 1 billion different receptors. Okay, so that seems simple, but how do we know that they're going to be different in you and me? So basically what we're going to do is we're going to take sequences that we get out of, as I described, out of the sequencing experiment. This is bulk DNA sequencing, but if you get enough sequences to do the analysis, you can do it on single cell too. We can talk, if anybody has questions about that, I'm happy to talk about it. I actually don't even see the Zoom bar at all. So if anybody, I guess you'll tell me if I need to pay attention.
Yeah, I'll let you know. Thanks.
All right. So, okay. So we get sequences out of a sequencer, and the name of the game now, what we want to understand is, what is the probability that an individual, say yourself, that you generated a given T cell receptor? Okay, and what is the probability of generating any given T cell receptor that we see in the data? And so we're going to use a trick. So basically, we need to quantify and characterize this process. So we're going to use a trick. And the first thing we're going to do is we're going to look at out-of-frame reads. So remember, you have 2 chromosomes, one from each parent. And when VDJ recombination happens, so this generation of diversity, first you attempt the first chromosome. If that works, great. That's the end of the story. But if it doesn't work, you silence it and you move on to the second one. If the second one also doesn't work, well, that cell dies. But if it does work, then you have a functional receptor. So you have a receptor that can be expressed on the cell surface and the T cell will survive. But then if you look in and you look at the DNA in the cell, you sample the DNA, you're going to find the two receptors. You're going to find the one that was a functional one, but also the one that recombined. The way these recombined but was not functional. So the way these experiments are set up is it's specific sequencing aimed at the immune system with primers designed for that. So you only sequence DNA that was actually recombined. But if you do sequence DNA, you're going to get these two chromosomes. So what we're going to do is we're going to use these out of frame reads. And since these reads were really, so basically the reads that come from this receptor that got recombined but was not functional and not expressed. So that way we're sure we don't have to deal with selection. And it's easy to understand why that's the case for DNA. Of course, mRNA, when I said it's silenced, it shouldn't be expressed. In practice, the process is leaky. And if we look at the mRNA data, we actually do see these out-of-frame receptors too. So we can also do this analysis based on cDNA sequencing. Okay, but we're going to use the out-of-frame reads. The important thing there is that we don't have to worry about selection. And then the second thing we're going to realize is if we take a given sequence that we find in the data, well, what we want to do is we want to say, what's the probability with which it was generated, which means we want to say, well, we need to say something, how likely it was that it used the given genes that it used as a starting point. But it turns out that if we want to assign which gene was used to the given sequence, deterministically, that's impossible. Because of this editing of inserting and deleting nucleotides, I can assign different V genes to any given sequence and different D genes, especially the D genes, which are quite similar and quite short. So we cannot do this deterministically. We're going to do it probabilistically. So we have to, we're going to make a list of all possible scenarios that are consistent with the sequence we see, by which I mean choice of V gene, D gene, J gene, number of insertions at each of the junctions and deletions. And we do it for all sequences. And we assign weights to each of these scenarios. And then we calculate marginals or observables, depending where you come from. Basically, we calculate the probability of using a given V gene in all of these sequences, having a certain number of deletions, certain number of insertions, and so on. And then we have a model, which is the most general factorializable model consistent with the known biology, which we use to recalculate these weights. And then we let this converge. So this is an expectation maximization procedure. That's basically how we learn the parameters of these models, of the model, which are these elements. And then once we have that, we're able to calculate these weights, so the probability of each of these scenarios. And by summing up over all of these weights, we're able to calculate the probability of generating a given sequence. So that way we can get the probability of generating a given sequence for every sequence we see in the database. Okay, so I won't tell you about the details of these models. I'll just tell you about the final outcome, which is what it looks like. This is the probability. This is a density or a histogram, if you want, of the probability of generation. So basically, we ask how many T cell receptors have a probability to be generated. That's 10 to the minus 10. And we put a dot here, 10 to the minus 8, and so on. And so there's two things to note here. One is that it spans 20 orders of magnitude, which means that you have some sequences which are very easy to generate, given that you're generating at least 10 to the 9 different receptors. A probability of 10 to the minus 5 means basically you will generate these receptors, and that means you will generate them and I will generate them and we'll share them. But these ones over here, they're much harder to generate, and we'll still generate some, but there'll be different ones that we generate. And it's thanks to the ones here that we're basically able to find a murderer. Because what this tells you is that each one of us is basically a random repertoire generator. We have the same, well, I'll tell you in a second why we have the same generation process. but we generate different realizations of the process, which in this case are receptors. So why am I saying that we have the same generation process? So the spread here, there's red, is over 20, basically is over 1,000 different people. So real people, not genetically identical, not mice or anything like that. And you see, and this is basically 95% variation. So you see that basically this process of generating receptors, this distribution is very, very similar in all of us. So it doesn't matter where you come from, doesn't matter what you eat. Basically, we have the same probability of generating given receptors. So yet, because it's a random generative process, we end up with different receptors. And again, these ones over here that have a high generation probability will share, and these ones will be unique. So there'll be some fraction that we share with other people and some fraction that will be unique to us. And basically, this uniqueness is such that we're able to identify any given person out of the whole worldwide population. So it's a unique fingerprint that you carry around. Now, at this point, you may ask, well, where does this distribution come from on evolutionary terms? And this is a question I would love to know the answer to. But one thing we asked ourselves is, well, does it depend, like if receptors that are specific and can recognize known viruses, are they more skewed to the right of this distribution? Do they have a higher probability of being generated? And the answer is no. So these are receptors, these are distributions over receptors that are known to recognize different viruses that are available in databases such as VDJDB, and we plot the probability of generation on top of the average that I showed you on the previous line. So there's nothing special about these sequences that are more likely to be produced in terms of them being good at recognizing bugs, basically, with, of course, the caveat that we're talking here about what's in databases. Going further into, is everyone the same? So when we did our murder mystery at the beginning, there's clearly a difference in samples coming from you and your identical twin if you happen to have one, but the identical twin still, was slightly shifted compared to unrelated individuals. So if we look at it in detail, they share a little bit more sequences than non-twins. And if we look at the PGen distribution, so basically this is the blue in model, red unrelated individuals, and the twins have this funny little arm here that that basically there's this long-term tail that they shared they're more likely to share these res sequences. So we looked into this in more detail, and so the funny thing is that what's so special about identical twins? So these are identical twins that share a placenta, and so basically identical twins that share a placenta are the only individuals in the world that share blood in natural circumstances. Not just maybe blood because you share blood with your mother, but share immune cells in natural circumstances. We're not talking about blood transfusions or transplants and things like that. And so they share blood. Whoops, question in chat. What is R in the previous plot? Oh, it's a correlation coefficient. Just the basically correlation, I think, between actually I'm blanking out. What is R? Confidence of these points being different from these points, I think. No, maybe, sorry. This is because I didn't go into this. This is the prediction of sharing in the model, and this is what we actually see in the data. And this is the correlation coefficient between what we see in the data and what the model predicts. Because originally this plot was showing 2 points that we're able to predict sharing based how many T cells are shared or between the model and the data, and that's the correlation coefficients between those. Sorry, that was a long time ago. So yeah, that's what it is. Okay, so good. Okay, so twins share blood, but they share blood before birth. They share blood in utero. And the other thing to know is that in utero, the enzymes that make the insertions, the enzyme called TDT, is severely downregulated. That means that the T cells you produce in utero are actually have many less insertions than T cells you produce after birth. So if we track the number of T cells with 0 insertions as a function of age, they go down. And we're able to fit a decay length to that, which gives you a clone lifetime of about 40 years, 36 years, okay? So this is a decay length of clones. It's not cells. Cells die, cells reproduce, but the clones that were generated in utero hang around for around 40 years. So that means that if you're about 40, you still have T cells that were generated before you were born. And this is not just due to the fact that the naive pole contracts, because that would be this decay curve here. So it is actually an observed decay of of the relative proportion of these 0 insertion cells, which allows us to estimate a lifetime. And we'll get back, Andrea will get back to estimating lifetimes later. Okay, so we already mentioned selection. So the first type of selection, so after generating receptors, they first basically undergo an exam, which we call, which is a way of thinking about thymic selection. So in the thymus, before they're let out into the periphery, they get asked 2 questions. Do you bind to anything at all? If they don't bind to anything at all, you're going to discard these cells because they're probably going to be bad receptors. But if you bind too strongly to self proteins, you're also going to discard them. So why is the thymus a good place to test this? In principle, the thymus is a pure organ and you should only have receptors that are specific to that are produced, sorry, proteins that are produced by your body. You should only have self proteins. So by basically being presented different proteins in the thymus, the immune system performs this discrimination tasks there on your own self proteins, and only T cells that don't bind too strongly to self but bind to some extent are let out into the periphery. And then they undergo somatic selection, which we already described, this proliferation for when they encounter an antigen, and otherwise death. So we can, for any of these selection processes, we can also build a probabilistic model and we can quantify selection. Basically knowing what is the probability of generating a given sequence. If we then look in the functional repertoire, for example, in the naive repertoire to quantify thymic selection, we can say that there is some selection factor which we can describe by some by a function. And this can be a linear model, such as shown here, where we basically say that selection independently acts on the V gene, J gene, and each of the amino acids in between, because this forms the CDR3 region, which is the most variable region, and often doesn't have, that's where all the insertions and deletions happened. Or we can build a neural network model, or any sort of machine learning model that accounts for nonlinear effects. And so we've done both of these. And, you know, there's small differences in essentially linear models, given the amounts of data that are available, usually do as well There are some things we're able to pick up with the with the neural nets, but it really depends on the deepness of the data. But at the end of the day, what we have is, since we've characterized generation and selection, we know the probability of seeing a given T cell in a naive repertoire, and then we can talk about start asking questions about functional repertoires. So the plot I showed you previously was an example of that, but we can ask it at the level of functional repertoires. We can ask how many cells are shared by 2 individuals, 10 individuals, 100 individuals, and so on. And we have the model prediction, which is basically the probability of the sequence being generated in one person and another. So say in you and in me, we generate these sequences independently. So that's just pgen squared times the probability that they were selected in each of us. Again, we're independent processes. So it's the probability that was selected in you and selected in me. And I can build a separate model for you, and I can build a separate model for me, or we can build a general model believing that thymic selection is still a somewhat universal process. Of course, there are differences for those of you that know about MHC presentation for T cells and so on. Either way, we can build a model like this, and we can see that the model predicts very well the sharing that we observe in the data. So the fact that you and I share naive sequences is still largely governed by chance for the most part in healthy people. Now, we talked about, I talked about because I'm the one talking, but I mentioned MHC. So T cells, when they recognize a protein, it's presented on the scaffold, which is called the MHC, or major histocompatibility complex. This is a scaffold that's encoded by the most polymorphic gene in the genome, the HLA. So it is a great source of diversity. So we are slightly different. So if we build a common model for people, we may need to include a sort of small selection factor that is person dependent or group dependent. And so basically, this will tell us that the higher the factor, the closer it is to one, the less stringent selection on a specific subsystem or subgroup of people, and the closer to 0, the more stringent selection. And here we looked at how it varies over different categories. So we can see that it looks different for people that are CMV negative or positive. CMV is a huge influence of how immune repertoires look like. And we see that it varies with age. So basically, as you get older, you have more signatures of more stringent selection in your repertoires. And so we can, again, go back. This is a different representation of what I was showing you about whether we can predict, and this is related to this this R factor that I showed before, which basically was measuring how well we could have predicted in that set of individuals we looked at, which is from a different sequencing platform than this. But you can see that we're doing very well at predicting sharing in healthy individuals with this selection factor for T cells. Same thing for B cells. We have smaller data sizes, but it works very well. But if we looked at people who recently had a SARS-CoV-2 infection, and this was people for whom this was a first infection, we see significant outliers. We see that these people share many more B cell receptors than predicted by chance. So this tells us that by looking at sharing in people, we're actually able to pick up some signal. And this brings me to the second part of my talk, where I want to talk about the fact that what we're interested in now is basically looking for specific signals in the repertoire haystack. So the repertoire is a very large system. some very small fraction, much smaller than drawn here, is used in a specific disease or condition setting, and an even smaller part of that is used in to identify specific antigens. And we would like to know precisely what this is, or more likely statistically what this is. So can we use this notion of sharing to try and identify sequences that are specific for given conditions? And the answer is yes. So here what we're showing is a prediction of how many TCRs should be shared between people and how many are actually shared between people in conditions that are diabetes and CMV infection. And the red dots, so this is an inverted version of the previous plot, with the red dots being significant outliers, and the ones in a circle are ones that were experimentally validated to be associated with this condition in previous experiments. So we can actually identify disease-related TCRs without having a negative cohort, so people who don't have this condition. If we go to back to SARS-CoV-2, from different data sets, we also have these significant outliers. We can identify motifs linked to them, and we can connect. This is for TCRs now, where we can also identify them. We can link TCRs that differ by at most one amino acid. And again, using VDJDB, we can link them to what SARS-CoV-2 epitopes they correspond to. And identify the logos and see whether the variability is. And if we go back to BCRs, which is by doing a simple log likelihood ratio test, we can identify whether basically looking at somebody's T cells, looking at the subset of them, we can say whether this person had SARS-CoV-2 or didn't have SARS-CoV-2 with perfect identifiability. For T cells, we need to do logistic regression, but by doing that, we're also able to do it. Another thing that it allows us to do is to HLA type individuals. So in short, just by looking at the T cells somebody has, and having learned a model, which I'll tell you about in a second, we're able to say what HLA type that person has. So what we did is we curated a database of T cells that are associated, that are strongly associated with a given HLA type. So HLA type plus negative donors for different HLA types, and from different data sets. And well, we confirmed that the strong association goes in the way we expect, meaning that HLA1 is mainly that CD8 is mainly associated with overrepresentation of HLA1 types and CD4 with HL2 types. The differences here are not errors, because we know that the same CDR3 and CD8 and CD4 can be associated with HLA1. The same CDR3 can be associated with CD4 and CD8. So this is not necessarily errors. It can be real. And then we build, based on this data, we build a logistic regression model to figure out basically what is the HLA type given on presence of absence of those strongly identified or associated TCRs. And you can see that whether we use just one change as the alpha or the beta, or both of them, we have very good performance. Now, what's interesting is that this is just saying that a given TC, we have to see an identical match in our database to in our individual to basically call an HLA type. Now, if we become a little bit less strict and say, what if we allow for a one amino acid mismatch in the TCR we're seeing in the person from the one that we know is associated to a given HLA type, it turns out that we actually do slightly better with this one mismatch was slightly above the diagonal, then we just allow for single mismatches. And so pushing this a little bit further, we can build clusters for different, different HLA groups of TCRs that differ by at most one amino acids. And we see that they cluster into these sets that then, through again VGDB, we can associate with specific known epitopes. So that tells us that similar TCRs have the same HLA restriction and bind similar or the same epitopes, which tells us that the information that is in a TCR repertoire alone can help us refine presentation models, but also binding models. So basically, there's a lot of information in TCR similarity, and it does seem to be an important element which we see over and over again, which we like to exploit. And so pushing with that, can we actually identify responding TCRs using just a single repertoire? And so to do this, and I'll show, we did this both on T cells and on B cells. I'll show you the B cell version just for a change. We can take a repertoire which has been exposed to a given antigen, and then we can build clusters of similar BCRs. And then what we do is we prune these clusters for what we expect based on PGen. Basically, we expect some similarity in the data. We expect these clusters to be there naturally because some BCRs have a higher probability of being generated, and they're usually ones with small insertions. So the ones that are short and with small insertions are more likely to be there. So we need to correct for that. But once we correct for that, we're able to actually use this as a way of identifying BCRs. So we took data from Steve Quake's lab, from Horns et al., and for people who received the flu vaccine at different types. And if we just look at frequencies or of counts of different BCRs, before and after vaccination, we don't see big differences. However, if we look at how basically how abundant the neighborhoods are, we see that after vaccination, we see larger neighborhoods, which means we see bigger clusters. So we can postulate that these larger or these BCRs that have more neighbors are potentially responding. And so we see this clear peak if we look at different individuals at day 7 post-infection. And in one individual, we can build a network of close by but unrelated sequences, meaning they use different V genes, even if they share similar CDR3s. And we really see that they have a different origin because although they share this, the ones that share exactly this specific CDR3 sequence, they're encoded by very different V genes that could not have been confused by chance. So we see convergence of a response. We see a convergent response of clones that were independently generated to the flu vaccines in BCRs. So I'm saying I'm out of time here. Should I stop?
We had some questions during the talk, so I think that's you can probably find to continue for a little bit. I think that sounds.
All right. So
and if we have more questions, I think we can go as we go.
Okay. So this is the last thing I want to say, sort of going back to this self versus non-self question. So that is presumably is decided during thymic selection. So the way, we like to think of it following Alan Perelson's shape space idea is that TCRs and pathogens live in the same abstract space. We don't know what it is. It's some sort of phenotypic space. And the name of the game for TCRs and BCRs as well is to cover the whole space. Because pathogens can be anywhere. But you don't want to put a TCR in a place where you have your own proteins. So these are sort of forbidden regions in black, but you need to cover all of the space, or all of this space. So in that sense, can we find features in T cell receptors that are able to discriminate foreign and self peptides? So in other words, can we find features in T cells, in T cell receptors, that basically tell us that this is a forbidden region? Are there some sort of features that are forbidden? I mean, maybe that's the simplest way of saying that. So we already talked about thymic selection, that you have to select for them being functional, so binding to anything, but self-tolerant, so not binding to your own self-proteins. And so this is a huge process that takes is very fast. It takes around 4 to 5 days. And so Arup Chakraborty back in the day did a calculation where basically the outcome of it is that there's not enough time for this. So given the number of self-peptides, if each T cell has to see all possible self-peptides, given the number of APC molecules, so antigen-presenting molecules, so the ones basically with the scaffold that present these peptides to T cells, there's no way that a T cell can see all peptides. It sees at best a few thousands. So it probes a small fraction of possible self-peptides. So remember, if this thing is set up in a way that you're supposed to actually make, a list of everything you're not supposed to recognize a T cell, there's basically no time to make the list. And the second thing is we know it's a leaky process. So Tregs are examples of T cells that bind to your own proteins. They go through this, so we know it doesn't work. And of course, we know we do get autoimmune diseases. So how systematic is TCR elimination? And again, do we see signals of this in the sequence? So to investigate this, we set up an experiment with Neo Friedman's lab in mice, where we had two markers, an activation marker, basically whether the cell is responding to something, and a cell death marker. And we sequenced T cells, not just in the periphery as we usually do, so functional T cells, but throughout thymic selection. So there's different cell types throughout thymic selection, and at each decision of whether the cell gets to proceed or whether it gets eliminated, we sequence the ones that passed and the ones that failed. So either you care about these different stages and you know about it, but essentially what we do is we have a bunch of examples that passed and failed different stages during thymic selection. And specifically, I'm going to really focus on this last stage, whether they led out of the periphery or whether they apoptose at this late stage. So what we're going to do then is we're going to build, again, a model, a selection model that takes us from generation to the given stage of thymic selection that we're looking at, to the given pass or fail. And we're going to build these models for basically the pass and fail groups. Now, if we just look at the data, even before learning a model, we don't see huge differences, say this is in V usage, between the different types. And again, the similar colors, like the pinky colors, are the ones, the lighter one is the one that passed, the darker one is the one that failed. Same for the orange. And basically, gene usage is really driven by the probability of generation. And there are definitely not differences between the pass and the failed ones. However, if we put all different, if we basically do a PCA analysis on the different cell types, they do cluster in a way that we expect. So first they're generated, then they go into this double positive, and they break up separately into CD4 and CD8. But again, the ones that pass and fail are very close together. And with these models, we are able to reproduce largely, again, the similarities that we expect from biology. So we are capturing something which is not capturing some sort of smoking gun. So we asked the question, but what do we expect? We basically, thinking about it as passing and failing, we basically expect that these two subsets should be mutually exclusive. So the energy functions that we learn from them should be anti-correlated, meaning that if I take a property such as length and I put a threshold here, then I call some of these T cells long and some of them short, and I build a separate model for the long ones and the short ones. Well, a T cell is either long or it's short. So if I then score all the cells with the short functions, the ones that were actually short will score high, but the ones that were long will score poorly with the short function, but well with the long function, and vice versa. So we're going to get an anti-correlation. However, even with such a strong anti-correlation, we may get a pretty weak signal of false positive versus true positive. Now, let's see what we get for this sort of energy anti-correlation on our past fail of CD4, CD8. And this is what we get. So this is CD4 here, the ones that failed, and the ones that passed and were functional. Same thing for the CD8. And we see that they're completely correlated. So it's the opposite of what we expect. Now, just to show that not everything would be correlated, the ones that die by neglect very early on, they're just the random subset of both the CD4s and the CD8s. So here we're really seeing that they seem to be selected for the same thing. And so maybe not surprisingly then, the area under the ROC curve is close to random. So this is for the CD4, CD8 choice. So basically, what does this mean? It means like if I have a bucket of cells, of CD4 cells that passed thymic selection and a bucket that failed, and if I put my hand in and pick one T cell at random and I'm asked, did I pick it from the bucket that failed or the bucket that passed? I can't really tell. However, if I now take 30 cells, so I do the same experiment, I put my hand in, but now I pick out 30 unique T cells from the same bucket, I am able to pretty well say whether they've passed or failed. And if I pick 100, I have perfect discriminability. So, and the threshold is around 30 cells, which is a similar order of magnitude that we've previously seen for T cell decision making in another context, in the decision whether to become a memory T cells, that was verified using microfluidics experiments by Michal Polański and Niels group at the time. So here they clearly showed there's some sort of collective decision making and quorum sensing rules. What we're showing with this is not that. We're just saying that in one TCR, there's not enough signal to decide whether it should die or live. But in 30 TCRs, there is. And so this is something we're trying to understand and we don't fully understand. This was so far done on single cells. On single chains. We separately did alpha and betas. The signal is the same. So maybe there is more signal in both, but that doesn't explain why we start having signal when we go to 30 even with a single cell. Anyway, that's the last thing I wanted to say. So probably the most important thing I want to say is don't kill anyone, even if you have an identical twin. This seemed to us like a sort of, fun twist, but it turns out that murderous twins is actually a serious problem in the world. And basically figuring out which twin did it. So people do care about that. I showed you how by looking at statistical properties in repertoires, such as overlap between individuals or in-host convergence, we're able to identify responding clonotypes. And Andrea will tell you how we can do this using longitudinal data. And for thymic selection, we're at a point where we were unable to find any deterministic rules for TCR rejection. So we believe there are some, again, either collective or statistical rules that we're interested in looking for. So thank you.
So it's a pleasure to give a seminar here. Okay, so yeah, I will present some like more details about quantification of the immune receptor dynamics. It's basically I will talk about three works done in the group of Alexandre and Thierry. I'm not the main author of all of them, but I'm quite familiar with those. And so in general, okay, the kind of Aleksandra already talked a lot about recombination and selection. But for us now, what's important is like after those processes. So in our body, then we will have like a pool of T cells or B cells with like a lot of different receptors. And to give some numbers, we can have like around 10 power 11 total cells in our body. And the diversity of these receptors, so in this case, the number of different colors it's not super easy to estimate this number, but it can range from 10 power 8 to 10 power 10. And so we are interested in understanding how this kind of composition changes in time and which kind of factor make those changes. Okay, so just to list a couple of like mechanisms that can change the repertoire composition. So of course, there is cell death, as Aleksandra said, and there is also influx of new cells through recombination and selection. And here I just mention, without entering too much into details, the fact that cells can also differentiate. So in the case, for example, of memory cells, they can take a decision of becoming memory cells that keep immunological memory, or to become cells that produce plasma antibodies. But there are also those kind of undergoing processes. And in addition, as also Aleksandra said, if like one cell in this repertoire encounter an antigen, a pathogen, which is close enough to its shape that is able to bind to it, basically this can start an expansion of the cell. And so the cell starts to duplicate and grow exponentially. And in the specific case of B cells, there can also be what's called affinity maturation. So basically the cell that's good enough to recognize the pathogen also starts to mutate the receptor. And the mutated receptor, after duplication, are tested against the antigen and selected for being good at binding the antigen. So basically there is a really like evolutionary process that creates better and better receptors. So we have like this collection of processes, and we would like to better understand like how these processes happen in healthy conditions. So basically, if we don't have any like measured disease, we can expect that also in healthy conditions, there are cell expansions because probably there are antigens in our body, and also affinity maturation can happen. Also, maybe those cells can react to the microbiome of our body. So we would like to understand how this happens in healthy conditions, and also what happens in response to a specific stimulus, and what really is different between responding to a specific stimulus and from the sort of like baseline kind of dynamics. Okay, and so this kind of idea can be schematized as follows. So you can imagine that in my body, as I told you, there are a lot of receptors, and you can like just count the number of cells having the same receptor. So I will call a color from now on a clone. So basically the clone, the purple clone has abundance count 3. And I can keep track of those numbers in time, and I want to study the variation of those numbers in time. And okay, so the kind of question we want to address are based on like understanding how this variation changes. But the problem is that like what makes this kind of inference, this kind of quantification hard, is the fact that I cannot really know these numbers, because actually what I can know are just sequencing experiments, so just subset of the real receptors. And again, to give some numbers, I told you, so if there are like 10 power 11 cells in a body, the number of cells that there will be in an experiment can range from 10 power 5 to 10 power 7. So you can really see that it's like really an infinitesimal fraction of the real number of cells. And in addition, experiments can introduce errors. There are amplification biases if there is PCR. So this like really makes this kind of quantification non-trivial. And as a matter of notation, from now on, I will call n prime the experimental counts, and n without a prime the real counts. And in general, I can write down a given probability of observing an experimental count given the real count, and I will call this the noise model. And I won't enter much into details of how those kind of noise model can be and how to learn them, but in general, they can be like, for example, a Poissonian model. So a Poissonian model would be like to model like a pure sampling. I'm just sampling receptors from this set. Of course, the probability distribution is a binomial, but can be well described by a Poissonian because of the big numbers. Otherwise, if like there are like amplification errors, people usually also use negative binomials that are like broader distributions. Okay, and in some also important fact, it's important to know is that the distributions of those numbers for T cells, for B cells, are power law. So here I draw actually a perfect power law. I don't really know if it's a perfect power law, but of course it's a distribution with a very fat tail with an exponent that's around -2. And like studying experiments that like basically are sampling from power laws brings some kind of problems, if you want. So basically, if I can imagine my experiment as a sampling from a power law, of course, I can characterize pretty well the behavior of the common clones because are easy to be sampled. There will be less error because they are more abundant, but I cannot know much about rare clones. And the problem is that in my case, also the majority of my clones will be rare. So basically, it's going to be hard to make an inference on the majority of the clones in my body. Okay, so these like are mostly general facts. I will like now go more into details and divide the presentation like in 3 topics. I will try to cover all the 3 topics. I don't know if I have time for the 3rd one, we will see. And so what make the topics difference is like the conditions in which we want to study the dynamics of the repertoire. So we will start from healthy dynamics. Then we will go to the response to vaccinations or infections in general. And the 3rd one is about the coevolution with the HIV. Okay, so considering the healthy dynamic, we can also have like a more specific question, more practical question that also biology are very interested in. That is the question of like how long I can expect a clone to survive in time. And of course, I mean, you can imagine those numbers changing in time. And in general, on average, you can expect a clone to decrease its number in time if it's not stimulated anymore. And so the question is how long I can expect this process to happen before extinction. And of course, this is connected also with the storage of immunological memory. So this is why like it's an important question. But so like a related question and sort of like more general question, we would like to have a model that is able to predict more this kind of variation of abundances in time in absence of like strong stimuli. So I will start by presenting like some sort of like qualitative approach that people already did several times in the past, that just allows us to say that it's true that actually the repertoire, the clones are quite stable in time. So the kind of like qualitative easy approach is the following. You can imagine that a patient is sequenced 2 times. And then what I do is simply just counting the number of clones I find at time 1, the number of clones I find at time 2, and the number of common clones. And I can easily then define like some sort of measure that like contains the fraction of common clones over the product of the number of unique clones at the 2 time points. So you can already see that actually those numbers are, if you want, sort of surprising. So if you really think that clones are very stable, you would expect to find many more common clones. So here the fraction of common clones is not very big compared to the fraction to the unique clones that you have. But like the reason here is that you have power laws, if you remember, and therefore when you sample, most of the time you will sample rare clones. It's very hard to sample the same rare clone twice. So that's the reason why your signal usually will be dominated by unique clones. But so you have this number here, and then you can like compare this number in the second with the second number. So basically you repeat the same procedure, but you do it with replicates. So like different experiments, but in the same biological conditions. You will get like, again, a fraction of common clones, and you can compare those 2 numbers in your, and you will see that actually it doesn't change much. So basically, if you consider samples after 10 years, it will be like more or less the same fraction of common clones that they will have between like replicates in the same condition. So this is really saying that the repertoire, in this case, it was like data from memory B cells. The repertoire is quite stable. So on a time span of 10 years, it's very easy that I keep my clone alive. So, but that's nice. We want to like improve a bit this kind of result. We want to be more precise, more quantitative. And to be more quantitative, we will use like a modeling approach. And so what we do is like imagining that my repertoire is evolving like a geometric Brownian motion. So I will like motivate in a couple of slides why I choose the geometric Brownian motion. Just let me describe how it is for those of you who are not very familiar with that. So it's a stochastic process. You will have here this eta is a Gaussian noise, and your abundance will evolve according to this equation. And you have to add like an absorbing boundary condition to like model the fact that the clones at some point will get extinct. So, for example, you can try to plot trajectories of this process, for example, here on the left in a logarithmic scale. So basically, this kind of process is special because if you do like a simple substitution of n being the equal of considering the variable logarithm of n, basically you get back a standard Brownian motion for which you can solve everything. So basically, this makes this process completely solvable. And so basically, if you observe this like kind of trajectory in a y logarithmic scale, basically this is a normal Brownian motion with drift that goes down. And so you will have this like exponential average decay with this time scale tau, and some kind of fluctuations that whose scale is set by this parameter theta. So on the right, it's exactly the same trajectories, but on a linear scale. And you can observe like this kind of like big fluctuations, giant fluctuations, that it turns out are important also because they are at the origin of the power law, as I will mention briefly. And okay, so and as I told you, like everything is solvable for this geometric Brownian motion, even with an absorbing condition. For example, one can compute the propagator. So it's the probability of observing a given experimental count at time 2, given the count at time 1. And basically, if you like you see the plot on the right. If you plot this on the variable log n, basically this is a Gaussian far away from the boundary. If you approach the boundary, it will change a bit the shape. But so in general, if I'm staying far away from the boundary, this is a simple log normal distribution for n.
I have a quick question.
Sure.
About the n=1 boundary condition. I guess maybe this model is maybe only valid for a subset of cells, like memory cells, as you were saying, because I feel like in many infections, you know, it's like one clone that's really expanding. So like at the beginning of an infection or something like that. Can you just comment a bit on, you know, what regime or like subset of cells this kind of?
Now, I mean, I'm really trying to model every cell in the body. And so I'm really saying, like, on a very simple level, I can imagine that whatever clones in my body will follow these dynamics. And at some point can get extinct if he hits the boundary n=1. But of course, I mean, these numbers can go up to 10 power 6, 10 power 7.
But it seems that, like, you know, most clones are unique to start with, and then they expand. So they
Can you imagine? Yeah, I mean, okay, maybe I'm coming this in the next slide, because I have to pair this kind of model with like an introduction of new clones with a given rate. So basically by mimicking the fact that you have an influx of clones. So every clone will start from a number that usually is quite small. But since I have this kind of giant fluctuation, every clone is possible to reach some kind of big level and then decay later. So, I mean, this kind of model is not really describing explicitly the expansions that happening because of like encountering the antigen or whatever. But, I mean, this kind of expansion are including this kind of giant fluctuation that can happen.
I see. So n0 is approximately what, like
it's order
2 or...
1. It's order. I mean, if I put
order...
exactly equal to 1, it doesn't really work, but it's order 1. Yes.
I see. Okay, yeah, then that makes more sense. Yeah, I guess it's just, you know, for like biologically, the intuition would be you start off with a unique clone, but you're saying that for this model to work, you kind of need something close to 1, but not exactly 1.
Yeah. Yeah, actually, okay, biologically, I don't really know what really happens close to the border. I mean, I guess, yeah. But yeah, I mean, the problem also is that, like, at the end of the day, we can really study this model through. I mean, what we can really say about abundances is through sequencing experiments, and I can sample only big clones, so I can really characterize what happens in the power law tail. I don't really know what happens in the rare clone regime, as I told you before. But yeah, okay. So like if I have like this model that like progressively like leads to extinction of clones because I have this negative decay on time, this negative exponential decay, and I couple it with like reintroduction of clones, at the end I have a stationary distribution that's a power law. And it's like the kind of one of main reasons why I choose this model. So the power law that's believed to be present in my repertoire. And also like a couple of other reasons is that it's simple and solvable. And the third reason is that there are also like previous works by Aleksandra, Thierry, that were like really studying more detailed models on how clones interact with antigens, expand and decay. And basically, if you take these models and you observe, like you study this model between 2 time points that are sufficiently far away in time, those models, they converge to the geometric Brownian motion. So basically, that's an additional reason. So you can see this model like as a convergence point for more detailed models. And if you look at your data between sufficiently separated time points. And by sufficiently long separated, I mean like order a few months, a year. Okay, so if like now we believe that this is the kind of baseline dynamics that we are assuming, Then we can try to include this like in an inference scheme. And so we imagine that my unknown real dynamics follow the geometric Brownian motion from one time point to the other. But as I told you, what I can observe are experiments. So basically, I can observe these n prime that are generated by my noise model. And given this setting, basically, I can write down the probability of observing experimental counts at time 1 and experimental counts at time 2 for a given clone I. And this will depend on some parameters that are the parameters of my geometric Brownian motion and the parameters of the noise model. So it's nothing very hard here. I just have to marginalize over the hidden variables. And at the end of the day, given this like probability, I can write down a likelihood. So the probability of observing my experimental counts at the 2 time points, and also by assuming that every clone is like independent in this case. And at the end, I can try to maximize my likelihood to get my best parameter that will like fit my geometric Brownian motion. Okay, this is like in principle doable. I just want to mention like a sort of a technical, important technical point. That's the following. So basically, if I try to perform this inference, this likelihood maximization by using only 2 experiments at different time points, basically there is a sort of like indeterminacy problem. I cannot learn at the same time the noise model and the geometric Brownian motion. So basically, I need data that have this kind of structure. So I need data that are at different time points, but I also need experiments that are performed at the same time. So I need the experimental replicates. And in general, I will need the experimental replicates to learn the noise model and then use the longitudinal data to learn the dynamical model given the noise model. So this is like a sort of technical point, but it will come back later also for the inference of responding clone. It's important to state it out. Okay, so this is like just to show some results. This was from work of Merriam of like a few years ago. And basically they are able to learn these parameters for different patients. So basically this plot, what is actually saying is that this line is the line that corresponds to a power law with exponent 2, which is what the people believe the repertoire sits in. And you can see that your data more or less are around this line. I don't really know. Actually, there is a group of points that's not super close, but it's probably a reasonable exponent anyway. Probably a more interesting like result if we look directly at the value of tau. So basically, we can really here see that my tau goes from 5 years to around 15, 20 years. So basically, this is really saying that the T-cell clones are extremely stable. So I remember you, this tau is the exponential decay time. So it really means that to see a decrease of a factor E, I have to wait 10 years. So if I start from a given abundance, after 10 years, the kind of like number of cells in the clone will be decreased of a factor 2 or 3. So it's a very, very slow decay. And also it's interesting to see that you can like plot this point as a function of the age of the patient. And you can really see that this tau increases quite a lot with the age. So younger patient, we have a faster decay. And you can basically try to connect this tau with the rate of reintroduction of clones. So if you believe that the total number of cells is conserved, you can basically compute what's the turnover rate, so the rate at which I reintroduce new clones, and basically it scales as 1 over tau. And so basically you can imagine that like as you age, your turnover rate slows down. So you will have like clones that live longer, but you have like also less new clones that will enter your body. So this is the kind of interpretation you can give to this plot. Okay, so that was about T cells. And briefly, I would like to also mention what happens to B cells. I just need like a very quick digression on how the B cell responds. And so again, we can start from like situation in which I have my pool of B cells. I'm calling them naive B cells because they are still not reacting somehow against the antigen. Basically, what like an antigen enters the body, there will be a good receptor that is able to recognize the antigen. And what happens is that on a time scale of some days, those naive B cells, they can differentiate into plasmablasts. And plasmablasts, they can attack the pathogen through antibodies. Otherwise, like a sort of different pathways is that like after weeks or months, those naive B cells initialize and carry out what's called an affinity maturation. So they start to duplicate, to acquire mutations, and the new receptors are selected to be good at binding the antigen. So we have like this Darwinian process that then will last for some weeks, some months, and will release in the blood memory cells and plasma cells. So actually my focus here will be on memory cells. So which is one of the main player for like keeping immunological memory. And like one little difference that there is with respect to the previous case of T cells is that like now here, instead of considering my set of clones, basically my basic unit contains all the memory cells that comes from the same affinity maturation process. So basically with the idea that like each affinity maturation is like responding against a given pathogen. And so all the memory cells that comes from that process will like somehow be in common and will be reacting against a given pathogen. So here is exactly the same from the mathematical point of view, like it's a set of numbers, but now my numbers is the number of cells coming from the same lineage, from the same affinity maturation. And then therefore I can like perform the inference as before, and I can get numbers as before. And I mean, surprisingly enough, basically I get very similar numbers as before. So I find that like memory cells lineages can have like this time scale of decay of 16 years for one patient and 13 years for the other patient. Also, the age of these two patients is quite old. I think one was 70 years old, the other one was 55. So again, this is like, it's very, very in agreement with the same result of T cells. Was not really expected from the beginning, but somehow we found similar number.
I have a quick
Yes.
question about that. Have you done experiments to see if these estimates are stable to sequence in depth? Because I'd be quite interested because you have the power law, so I don't really know how that.
Yeah. Okay. I mean, in principle, all the kind of framework includes the sequencing depth in the picture, so it shouldn't be independent at all for the sequencing depth. I'm pretty sure that like in the case of Merriam, for example, in this other case of T cells, I guess like each kind of point. So you can see, for example, you have patient 4 that is like yellow, and you can see different points. And each point is a different pair. So you have multiple time points and you perform the inference between every pair of time points. And therefore, every experiment will have also different sequencing depth. And the fact that you obtain similar values like should point towards the direction that this kind of procedure shouldn't depend on the sequencing depth. I don't know if I answer your question or.
Yeah, I think so. But I guess there's also the even with the same pair of samples, if you just downsampled the.
Okay, in that sense. In that sense, I mean, I really think nothing changes. I don't know if they tried in this work. What I can tell you also is that like those numbers have been obtained from bulk repertoire sequencing. So where the sequencing that was like, I don't know, I think the number of cells in an experiment is around 10 power 7, something like that. These numbers for B cells have been obtained on single cell datasets, where the sequencing that is like 10 power 5, so it's much smaller. But yeah, I mean, I really think that nothing changes just by downsampling. I would find exactly the same. Of course, if you downsample too much, then you start to lose signal, and probably you will have like a larger error bar that your inference find. But yeah.
Okay, cool. So it's pretty robust.
Yeah, no, I'm pretty sure it's quite robust. Yeah. Okay. Okay, so like to wrap up this part, so we have like a sort of framework that capture the statistics of the receptor abundance dynamics in healthy condition and accounting for experimental sampling. Like we can try, this kind of framework can also like answer the question of like about repertoire stability, which we find to be like surprisingly long, and potentially can give us like predictions about future experimental counts. But so like to like point out some limitations of this kind of framework. So as I told you before, we are trusting a geometric Brownian motion, and probably this kind of model describes well the dynamics only if your like experimental time points are sufficiently far away in time. So if you look at the dynamics after a few months, probably the model won't be able to capture well the kind of statistics. And another kind of like limitation can be the fact that I'm really treating every clone exactly with the same parameters. So I have a single geometric Brownian motion that describes every clones in my body. This is probably not really realistic. So probably there will be like clones or like groups of clones that will die faster, group of clones that will stay longer. Also like to like trying to match the kind of expectation we have. I mean, the kind of knowledge we have about like immunological memory. We know that for some vaccination, the immunological memory is very long. For some other, the immunological memory is extremely shorter. And so basically, we believe that at this point, we are not really able to capture this kind of variation that we have across all the different lineages we have in our body. Okay, so the second part is about like understanding what happens in response to vaccinations or infections. So the kind of situation now is the following. So you can imagine that when you encounter a pathogen or an antigen given by some vaccination, a few of your clones will start to expand. So through this kind of exponential duplication. And at some point we'll start to contract, coming back to a given baseline. And the question here is really trying to understand by looking at those like trajectories, so experiments performed at multiple time points, trying to understand which are those clones that expanded. And again, like the kind of difficult part to do this kind of task is that in experiments you have like, again, experimental noise. And so you would like to disentangle between the real expansions and the noise that the experiment generates. Okay, so like a first approach is based on a previous work of Aleksandra and Thierry, is on a data set where those two people were like infected by COVID, and they basically collected samples of the T-cell receptors starting from the 15th day after infection up to like 3 months after infections. And there are 5 time points. Basically then for each clone, you can imagine that you can trace like a trajectory of abundances across these 5 time points. So here I'm just plotting the very big clones, because you can imagine that a lot of clones will be 0 in a few time points. So this is like the top clones. And what you can do here, like by looking at those trajectories, is to imagine those trajectories to be like vectors in a 5-dimensional space. You have these 5 time points, and you can try to run PCA on this space. So this is a very simple idea, but it works surprisingly well. So if you do this, of course, I mean, you need to like do some sort of pre-processing on your trajectory. Basically, you would like that all your trajectories, the variations of all your trajectories are comparable. So basically, you have to rescale, normalize all your trajectories. And what they do in this work is just dividing each trajectory by the maximum point, such that all the trajectory is like bounded between 0 and 1. But like modulo this kind of processing, pre-processing, you run PCA and you find like really different clusters, different behaviors in the, for example, here in the two principal components. And then if you like plot the kind of average behavior of the trajectory in the different clusters, you really observe like different trends. So in the violet ones will be the constant trajectories. So probably trajectories that are not caring at all of the presence of COVID. Then you will have like these green ones. So, and after day 15, basically you're observing that they start from a big count and they decay. So they are contracting. So therefore it's very likely that they responded to the disease and now they are just contracting and returning to the baseline. And then also you observe like a third cluster that somehow it seems to correspond to a secondary response. But I mean, then in this paper, they really went more into details and tried to like verify that the clones they observe in this yellow and green cluster are effectively clones that are able to bind to COVID, and it seems so. But like I would like just to point out to the fact that the method is very simple and is able to like capture some kind of statistics and isolate different behaviors of your trajectories. Also, this is saying that it's also capturing the fact that, like, as people believe, like T-cell clones, typically they have like an expansion time scale of like a couple of weeks. And so if you look at the green clones, probably those are really the clones that expanded at the maximum point after 1-2 weeks, and now they are decaying. But okay, this like was like a first approach, but actually if you want, this kind of approach is not really possible to being used like in a systematic way, because there are like different choices that you can do a priori that are not very well established. I mean, you can take different choices and obtain different results. I mean, one choice is the fact that I have to normalize trajectories. Is not that there are many choices of doing this. I can consider counts or log counts, but probably the biggest problem is that here I'm not really taking into account in a systematic way about the noise. Basically, what I'm doing here, so in this kind of example, basically in this paper, they were selecting the top 1,000, if I remember, clones in the dataset and performing the analysis on this top most abundant 1,000 clones. If you repeat the analysis by choosing like the top 10,000 clone, you stop to see any signal, because then at some point your signal is dominated by noise. So there is not really like a threshold, a number that's valid for every experiment that tells you to choose like a given number of top clones to perform your experiment. So it doesn't really quantitatively take care about the experimental noise. So like to include a more quantitative approach to like model response in presence of experimental noise, we can use like an approach similar to the one we used before. So this was exactly the same picture I showed before for the geometric Brownian motion. Instead, now, of course, as I told you, geometric Brownian motion was working well in healthy conditions for very well separated time points. Here, I want to change my model, my dynamical model, by including something that models these kind of expansions or contractions that I'm observing in my clones. Okay, so let's try to look at this model. This model is the following. It's quite simple. So basically, it's I'm doing this kind of introducing this new variable, this S, that is the log fold change. So basically the logarithm of the ratio between the counts at time 2 and the counts at time 1. And I can express, of course, N2 as a function of this S. And my probability depends on this S. So we will have like two terms. Let's first focus on the terms on the right. So basically you have this F, that's the fraction of clones that you are believing are expanding. So they are reacting to the disease, to the vaccination, and are expanding. And this F multiplies this rho, which is a probability distribution over the log fold changes. So it's a sort of, if you want, prior on what would be the distribution of log fold changes that your dataset will have. Actually, the kind of results of the inference don't depend too much by the specific choice of this distribution. And then you will have a second term that's with probability 1 over 1 minus f. So the fraction 1 minus f of your clones will not expand, will not contract, and basically will keep their log fold change equal to this S0. So this is a delta function. And this S0 is a number that's very close to 0, but it's not exactly 0 because basically I need to like conserve the total number of cells in the body. So if some of the cells are expanding, all the others, they have to contract a little bit. So that's the model. So I will have like a couple of new parameter, F and S0, and typically I have like one parameter more in my distribution of log fold changes. But the idea now is like exactly to do as before. So I can write down probabilities of observing experimental counts, writing a likelihood, and trying to maximize the likelihood. Okay, so I can do this. I can obtain like the best parameters given my experiment. And what is actually interesting, the way this kind of framework can be used to like identify expanding clone is the following. So I can compute a posterior. It's basically a probability of observing a given expansion size, given some experimental counts. And this kind of posteriors can be easily computed given the probability I showed you before. And then at the end, I can use this posterior as a sort of statistical test to identify expanding clones. So basically, I can compute what's the probability of a clone not to expand, so of having a log fold change smaller or equal to 0, given some experimental counts. And if this probability is smaller than a given sort of statistical confidence alpha, I can say that those clones with those experimental counts are likely expanding. And so I can select them and they can be like candidates for like future investigations. And so like as just like to visualize a little bit what happens if you plot in this like kind of diagram the counts at time 1 and the counts at time 2. This kind of condition with this confidence alpha, basically it will trace a line on this plot and all the points above this line. So with a count at time 2 that's much larger than what would I expect if there are no expansion is set. Basically, this gives me all the clones that are probably expanding. And so this kind of procedure has been discussed in a paper of Alex and Thierry, and also has been like translated in a software. So there is this open source software that is able to perform this kind of Bayesian analysis to identify expanding or contracting clones in data. And so like to conclude this part, I told you that we have like this framework that is able to disentangle between the experimental noise and to identify like responding clones. It's a Bayesian framework, so in principle there is also room to play with the prior on the log fold changes distribution. And starting from like this framework, there is also a software that can be downloaded and in principle is like easy to use and can be used on real analysis. Again, I would like to mention a limitation, which is the fact that actually, as I told you before, again, I have a noise model entering in my likelihood. So I need to like learn the noise model before, but to learn the noise model, I need experimental replicates. So basically the main limitation of using this kind of tools is that I need 2 time points and 2 experimental replicates to perform first the inference of the noise and then the inference of the expansion. But then if those are there, basically all these kind of frameworks should reliably take into account about all the kind of problems that can be in the errors in the experimental case.
Hey,
Okay,
Andrea,
so
before you go
again-
to that part, just a really simple question. Like, how does the Bayesian approach compare to like the naive approach that most people use, which is just looking at clone frequencies at time point T1 and T2, and if it
Yeah, I
goes up
mean-
over a certain amount, like how many, can you just say something like, maybe I missed it, but like, can you just say what you think the false positive and false negatives we're getting when we just do a very naive
Yeah, okay.
approach?
I think by better learning what's the kind of noise you would expect, basically your model would like change a little bit the threshold, so it would shift a little bit this line. So you can say, okay, I want to choose with confidence 1% the contracting clones. If you would use like a standard model that doesn't account properly of the experimental errors, you would always choose the same threshold. This kind of approach, we would move the threshold depending on the intensity of the noise that it finds somehow. So in an experiment with a lot of noise, it will choose less points, and probably that's the right way to do, because you are less confident because the noise is stronger.